In lecture 4, we're going to be talking about how we use DNA as a blueprint in order for us to produce proteins that are necessary to run all the functions in life. And we're going to also tie in the genetics of cancer as a backdrop to kind of kick things off. So let's get started with lecture at 1, how can a gene mutation lead to cancer? All right, the first thing we're going to do is we have to review what a gene is. So what I have here for you folks is our strand of DNA, and we can now, from DNA as the blueprint, make a copy of it. That's our messenger RNA. And later on ribosomes, we can take this mRNA, which is basically directions on how to make a protein and then make a protein on ribosomes. So proteins are literally the machines that are going to run our life. Okay. We can't live without these proteins. They are our receptors that hormones bind onto. They are enzymes that allow us to build molecules in our body or break them down. Okay. Now, in DNA carries a lot of genes. So a gene, singular, is a specific portion of the DNA that is going to code for a specific sequence of a polypeptide. And a polypeptide is just a long protein chain, just like here, right? This is amino acid, amino acid, amino acid, amino acid, and later it's going to fold up into a huge protein. So some proteins may consist of many polypeptides, okay? So if that's the case, in order for you to make some, let's say you need a receptor for the insulin hormone, and maybe it takes 3 polypeptides that come and wrap themselves up together, then that means you need 3 different genes to code for that insulin receptor, okay? Now, a single chromosome then can contain thousands of genes. And you don't need to memorize these numbers, but I just want you folks to get a deeper relationship of what it means by this word gene, okay? You folks good? If not, make sure to type in your questions in our virtual office hours. Now, the second thing I want to do is I want to review genetic regulation. And we have had this in a previous lecture, but you know, this is a new topic, and because we can't see it with the naked eye, sometimes it can get a little esoteric. So let's review it again. Now, remember that proteins, all proteins are coded by genes, okay? And we need proteins to bind to our DNA in order to turn protein synthesis on and off. Remember, we have to be able to control life, and we do it by the ability to turn things on and turn things off. All processes in our body has to have an on switch and an off switch, much like the lights in your house, okay? Now, upregulation. Remember, upregulation means we are going to now make more proteins. And remember, in our macromolecule lecture, proteins is a whole broad array of proteins. But when I'm talking about proteins right now, we're talking about functional proteins. Yes, receptors, enzymes. Now here, what this picture is showing you is if you have now some ligand, a protein ligand, and it's bound to the promoter region, okay? So remember, when things bind to the promoter region, then you can start upregulating that gene to make proteins, okay? Now, look at the picture, okay? Observation is important. How many ligands are here? I got 1, I got 2. So I can maybe make this protein, if this is gene 3, I can maybe make protein 3 at a very small level, okay? Maybe I need some enzyme and it's being made a little bit. Let's give it a number, 10. Okay, I made 10 proteins. But when I start to get more of the ligands, right? So this is 1, 2, 3, 4. then look, it really bumps up how many proteins. In this case, this is going to be protein 3. Maybe now I can make 100 of them, okay? Is because the conformation, which means shape, the conformation of these ligands and how they now bind on the promoter region will recruit more enzymes in order for us to undergo more transcription of this gene. That's upregulation. Now, remember, in order to sustain life, we have to be able to turn things off, and that is downregulation, right? And so here, what you folks are seeing here is there's a ligand right here, and a ligand that turns off gene transcription or starts downregulation processes is called a repressor, okay? And then what it does is when it binds to the area by the promoter region, it's called the operator region. But if you say it's in the area of the promoter region, that's fine too. But when it binds, what it does is its specific shape blocks this enzyme that allows us to make our mRNA molecule. And in doing so, we stop gene transcription. And that's how we downregulate. Everything in this course moving forward, all of the lectures are all about the shape of a protein and how different proteins come together and bind in order to do some function. And if you don't have the correct shape, you stop function. That is, if anything, one of the most important concepts you need to understand to grasp everything in this class. So to sum up, gene regulation, okay? These are all on this slide, mechanisms of how we can now turn on certain genes, but then turn off other genes, okay? All by ligands and their specific shape. All right, now let's get into gene mutations in cancer. So first, I want to define cancer. And in a nutshell, cancer is unchecked cell growth. What does that mean? That means that the cells have lost the ability to downregulate genes that code for protein ligands that control cell mitosis. Okay, so now all these cells are dividing uncontrollably, right? Now, most mutations leading to cancer are acquired and few are inherited. So what that means is majority of cancers are caused by mutations that are acquired throughout our lifetime based on our exposures to something called mutagens, right? So for example, mutagens, and I'm going to talk about mutagens later on in this lecture, but mutagens are like, for example, UV sun rays, okay? That's a mutagen. Certain chemicals like formaldehyde, those are mutagens, is because they're known things that can cause a mutation in our DNA. Now, there are two types of mutations that can lead to cancer. And I want to state something first, because this is an important concept. But not all mutations lead to cancer. Okay? Majority of the mutation or mutations that we acquire throughout our lifetime, they're called silent mutations. Okay? More on that later. So if you get a mutation in your DNA, there's two types of mutations that do lead to cancer. And the first one is if you get a mutation in your oncogenes. And so if you get a mutation in your oncogenes, it's analogous to as if you're in your car and your car moving forward is representing cell mitosis. The cell is dividing, okay? Now, imagine that your gas pedal is stuck down and you can't control that. It's just, That's cancer, okay? Now, the other type of mutation you can get is in your tumor suppressor genes. And the tumor suppressor genes are genes that code for proteins that help you stop mitosis. So if you mutate them and you no longer have the ability to brake your vehicle, then you're also going to be driving very, very fast, okay? So let's take these mutation types and talk about them more in detail. All right, let's talk about oncogenes. Now remember, oncogenes is when our mitosis vehicle is driving way, way, way too fast, okay? A lot of mitosis. So here, proto-oncogenes, they code for proteins. So remember, ligands, right? Genetic regulation ligands. They're going to code for proteins that stimulate cell division. So these things are usually called growth factors, right? And so growth factors will regulate how often the cell will grow or divide, right? Now, if you get a mutation in the specific area of where the proto-oncogenes are in DNA, then we call it an oncogene, okay? So oncogene, bad. Proto-oncogene, good. Okay? You folks got this? Now, if you start getting a lot of mutations in the areas that code for these proto-oncogenes, the more oncogenes you get, that's going to cause or lead to an increase of the number of proteins or growth factors that you're going to start to make. Okay? Now look, remember this picture? On the left, I don't have a lot of these proteins to upregulate my gene transcription. However, if I start having a bunch of oncogenes, I'm going to start making more and more of these protein ligands that upregulate gene transcription. Then I'm going to get into trouble, okay? And so here, what can happen is I'm going to have mitosis permanently turned on. That's unchecked cell growth, which is cancer. And then you can also have mitosis activated when it's not supposed to be. All right, now let's talk about tumor suppressor genes. So generally speaking, the job of tumor suppressor genes are to code for proteins that do the following functions. They're going to slow down cell division. They can repair mistakes in the DNA. So later we're going to talk about how we replicate DNA, and yeah, it's really easy to make a mistake. And any mistake that's made via the cell or because a mutagen has hit the DNA, those can be repaired from proteins code from these tumor suppressor genes. And then ultimately also is there are proteins from these specific genes that signal the cell to undergo something called apoptosis, which is signaled cell death. And apoptosis is important because as cells live, they will accrue mutations. And so a cell can only live for so long is because its function also starts to deteriorate. So you kill those cells and you renew or regenerate new cells to take its place. Now, there's something called breast cancer genes, commonly known as BRCA, and we have two of them, BRCA1 and BRCA2. Breast cancer genes do not cause breast cancer. Breast cancer genes are tumor suppressor genes, and they specifically code for proteins that suppress cancer, okay? Now let's talk about BRCA. And BRCA is very popular in the news and in the media. And the BRCA gene, so remember, there are many genes on a chromosome, and BRCA1 is on chromosome 17, and BRCA2 is on chromosome 13. And we have 2 copies of the BRCA genes. Now, don't memorize the chromosome number and whatnot. This is not a genetics class, but I just want to show you folks these genes is because I want to introduce what it means to have mutations or give you a specific example of having a mutation in a tumor suppressor gene. Now, if you have inherited mutations in your BRCA1 and or BRCA2, you are at increased risk of breast and ovarian cancers in women, and also other non-reproductive cancers. There's an increased tick in pancreatic cancer, colon cancer. And for guys, same thing. If you have an inherited mutation, you are also at risk for male breast cancer, prostate cancer, pancreatic cancer, melanoma. Okay. So this is not only geared towards female, male and female. If you inherit a mutation in this specific tumor suppressor gene, you are at an increased risk for different types of cancer, especially reproductive cancers. Okay. Now, if you do inherit mutations, you have a 25% hereditary increased risk of breast cancer and 15% of ovarian cancer. And you folks don't have to memorize these percentages, but I show you folks this is because, do you get an increased risk? Yes, you do. However, there's a lot of environmental factors that also play into whether or not you develop cancer at a later age, right? Diet, right? How you live your lifestyle. If you're a cigarette smoker, what toxins are you taking into your body? How much sun do you get? Or do you go in those tanning beds, etc.? Okay. Now, if you do get breast cancer, ovarian cancer, and if it is associated with BRCA1 and or BRCA2 mutations, usually you develop cancer at a younger age. Which brings me now back to this Angelina Effect article, because Angelina's mom died very young, and she died very young of both breast cancer and ovarian cancer at age 56. And that is very young to have cancer. Usually cancer can develop later in life, because cancer is an accumulation of your mutations and your lifestyle, right? And so because she now got tested for the BRCA gene and found that she had mutations in 2013, she had elective double mastectomy, right? After she found out that she had mutation in her BRCA1 gene. And the doctors, based on her genetics and her history and her mother's history, said that she had an 87% chance of having cancer. And so for her, especially as a high-paid actress, especially where breasts are important, having a double mastectomy and taking our breasts out created this huge ripple effect at the time, and got a lot of awareness about the BRCA gene and testing, especially genetic testing for women. So here I have some YouTube clips. If you're interested in watching, it's not necessary. I do get a lot of questions about BRCA genes, and so here are some recommended watch videos that you can watch that talk about BRCA genes and cancer, and then also, should you get tested for the BRCA gene? Some people don't want to know. They just want to live their life. And we don't judge, okay? There's no judgment, and that is a personal choice. All right, that's it for this lecturette. I'll see you folks for lecturette. What are we on now? I'll figure it out when I press pause. All right, bye.